Got healthy mitochondria? The key to understanding Parkinson's. If you are here at the World Parkinson's Congress, the most important question to ask is, do you have healthy mitochondria? Our late-breaking poster presentation at the 2026 World Parkinson's Congress highlights how recent advances in understanding mitophagy, the cellular process of selecting and recycling old mitochondria, offer fresh insights into the onset and treatment of Parkinson's disease. What are mitochondria? We breathe in oxygen in our lungs. Red blood cells carry that oxygen from our lungs to all our cells. Oxygen enters our cells, and then where does it go? It goes into tiny organelles called mitochondria. A major job of mitochondria is to produce energy in the form of ATP. The oxygen we breathe in is used inside the mitochondria. Food is broken down into simpler fuels such as amino acids, fatty acids, and carbohydrates. These fuels enter the mitochondria, where they combine with oxygen to produce water, carbon dioxide, and ATP. The carbon dioxide we exhale comes from the mitochondria, which are always busy making ATP. We have many thousands of mitochondria in a single dopaminergic neuron. Mitochondria live only for a few weeks. When mitochondria wear out, they must be recycled. For every mitochondrion that is recycled, a new one must take its place. Mitochondria are replicated by dividing into new mitochondria. There is more to making new mitochondria than dividing by fission. The whole process is called biogenesis. Mitochondria import most of the enzymes they need after they divide. Mitochondria fuse to form networks and exchange parts. Mitochondria are also carried back and forth in the axons or branches of the nerve cell. If all goes well, you have healthy mitochondria. If the thousands of mitochondria in a single nerve are damaged and not being recycled, you have Parkinson's disease. That is the theory of our poster presented at the 2026 World Parkinson's Congress. So, our poster was accepted as a late-breaking poster for the World Parkinson's Congress. That was great. I have been going to the World Parkinson's Congress since my own doctor, Lisa Schulman, helped organize it and invited me to the inaugural Congress in Washington, D.C. I came up with the seed idea for our poster during a poster tour in Portland, Oregon, where I met Colby Samstag, who was then a graduate student. His poster was looking for the mechanism that identifies unhealthy mitochondria for recycling. My name is Bill Curtis. I have had Parkinson's disease symptoms that were obvious since 2000. I recently co-authored a paper with Patrick Bradshaw on how mitochondria are selected for recycling. The poster is based mostly on that paper published in Redox Biology this past January. Dr. Seeds also helped with the poster. He is a medical doctor who co-authored 2 previous papers with us. When we made the poster, I was hoping he could give a hot topics talk, but that did not work out. I brought this poster to my Parkinson's support group. I wanted to see how they would respond to it. Only the people sitting next to me at the table looked at the poster. No one had questions about it. I was a little bit disappointed in their response. When I looked at the poster, I did not see a single point we had made that a person with Parkinson's disease or a caregiver could understand and relate to their experience of Parkinson's disease. I had failed to communicate. I came home and wrote down these questions that I thought the poster answered. I chose questions relevant to people with Parkinson's disease, as well as to caregivers, doctors, and researchers. I came up with these questions. 1. Can you explain how infection, inflammation, trauma, alpha-synuclein, environmental toxins, and genetic risk factors converge on a single mechanism of Parkinson's disease initiation? 2. Do you know how exercise improves Parkinson's disease symptoms? 3. Do you know how unhealthy mitochondria are selected for the recycling process called mitophagy? 4. Do you know how mitophagy is linked to the formation of new mitochondria? 5. Do you know how to make your mitochondria healthy? My goal in this presentation is to introduce the poster using these 5 questions. At the end, I will point out 2 more questions that researchers need to understand to comprehend the poster, but I want you all to stay for the first 5 questions. The poster has 3 major sections. The central section uses a single illustration to explain the steps involved in selecting unhealthy mitochondria for the recycling process called mitophagy. The steps show the perspective from inside the mitochondria. The steps are numbered, and the explanation of each step is found in the text of the center section. Here, we also note that mitophagy is linked to mitochondrial replacement. If we look at the panel on the left, there is an introduction that asks, is Parkinson's disease caused by a loss of mitochondrial quality control? Here, we recognize that the designer drug MPP+, which was the result of an unsuccessful attempt to synthesize a recreational drug way back in the late 70s, caused PD-like symptoms that responded to L-DOPA. The MPP+ is driven into the mitochondria by its positive charge. The healthier the mitochondria, the higher the gradient of charge across the inner mitochondrial membrane, the more likely MPP+ will enter the mitochondrial matrix. So, MPP plus causes the removal of healthy mitochondria. This shifted the proportion of healthy to unhealthy mitochondrial populations, allowing unhealthy mitochondria to dominate. Having fewer healthy mitochondria brought on severe Parkinson's disease symptoms. If the recognition mechanism that selects unhealthy mitochondria for removal is damaged, the population of unhealthy mitochondria will build up in our neurons. The removal of unhealthy mitochondria occurs regularly to maintain a healthy mix. There are several thousand mitochondria in a single dopaminergic neuron. If the mechanism for detecting unhealthy mitochondria is disturbed, it has the same effect as MPP plus on mitochondrial populations. Failure to remove unhealthy mitochondria allows the pool of unhealthy mitochondria to increase. It is reasonable to expect that failing to select unhealthy mitochondria for mitophagy would result in PD symptoms. Next comes a key point in the poster. Scientists don't know how mitochondria are selected for mitophagy. Our theory claims to answer the question, do you know how unhealthy mitochondria are selected for the recycling process called mitophagy? Also, in this panel on the left, we claim that several diverse PD initiators all have a mechanism that interrupts the selection of unhealthy mitochondria. The logical conclusion one can draw from this is that if a person is exposed to any of these initiators, they can develop Parkinson's disease. This is a monster claim. We are saying that this is a cause of Parkinson's disease. I would argue that it is a more useful explanation than explanations that fail to explain how all these initiators could cause Parkinson's disease. The panel on the right suggests that if the theory of how mitochondria are selected for mitophagy is correct, then we can propose a treatment based on optimizing the selection process. The poster lists several vitamins, minerals, and nutrients that are needed for the selection of unhealthy mitochondria for recycling. This section suggests why exercise works and suggests what types of exercise work to give you healthy mitochondria. This is where I switch to explaining two questions that are for researchers. You are welcome to stay, but it gets more technical. I had two more questions that I removed because they concerned ideas hardcore researchers have stumbled over. 6. Do you know why oxygen is paramagnetic? Paramagnetic molecules have unpaired electrons. If all the electrons are paired, the molecules are diamagnetic. 7. Do you know how NADH and NADPH can have different redox potentials in the same membrane-bound compartment? Why is oxygen paramagnetic? Why do I draw the molecular form of oxygen with two dots? The two dots indicate that oxygen O2 has two unpaired electrons. Scientists are generally unfamiliar with this fact, and everything they do know about molecules with free electrons or free radicals suggests that it is a ridiculous idea to say molecular oxygen has two free electrons. Free radicals, which are familiar to them, cause uncontrolled chain reactions that tear apart biomolecules. How could this lack of knowledge be so prevalent? I knew oxygen had two free radicals because my chemistry professor, Professor Herlocker, had let me into his course on chemical applications of group theory when I was a freshman at McDaniel College. Group theory is a branch of mathematics used to solve problems in quantum mechanics arising from molecular orbitals. Later in graduate school, I took a course on the history of modern physics taught by Stephen Brush at the University of Maryland in College Park. Brush had written a treatise on two competing models for understanding molecular bonds. Valence bond theory used valence electrons, and molecular orbital theory used quantum mechanics. In valence bond theory, you represent an oxygen molecule using Lewis dot structures. The dots represent valence electrons. Dots are much easier to understand than this diagram, which shows three states of the dioxygen molecule O2 in the molecular orbital model. I found this quote from Dr. Brush's treatise regarding the impact of textbooks such as Linus Pauling's book, The Nature of the Chemical Bond. Here's the quote. In his review of Nature of the Chemical Bond, Joseph Mayer, husband of Maria Guppert-Meyer, worried that because of the excellence of its presentation, valence bond would eclipse molecular orbital, even though the latter was needed to explain some phenomena such as the paramagnetism of oxygen. Confirming Mayer's fear 30 years later, Huckel complained in his memoirs that, by delaying the acceptance of MO, this book retarded the progress of quantum chemistry for 20 years. In his book The Double Helix, Watson mentions that Linus Pauling's Nature of the Chemical Bond was used to understand how hydrogen bonds match the base pairs in the double helix structure of DNA. So, if you are not familiar with quantum chemistry, you will have to trust us that oxygen is a dual free radical. The major takeaways about quantum chemistry are stated very simply in the explanation of the steps in the center section of the superoxide sentinel theory. The seventh question I bring up is because a reviewer once denied our premise that NADPH and NADH have different redox potentials. If you look them up in tables, NADH and NADPH do have the same redox potentials. This is true, but in life, NAD+ behaves more like a dead battery waiting to soak up electrons. NADPH functions more like a fully charged battery eager to donate electrons. One needs to charge and use fully charged forms simultaneously in the same compartments of the cell. NAD+ and NADPH can maintain separate redox potentials within the same membrane-bound spaces by using the presence or absence of the phosphate group to enable recognition by different sets of enzymes, thereby determining their reactions. This was first discovered by our late mentor Richard Veitch when he was working in Hans Krebs' laboratory. That is all I wanted to say to introduce our poster. If you have questions, put them in the comments, and I will try to answer them.